Chapter 42 The Immune System Figure 42.1 In this compound light micrograph purple-stained neutrophil, upper left, and eosinophil, lower right, are white blood cells that float among red blood cells in this blood smear. Neutrophils provide an early, rapid, and nonspecific defense against invading pathogens. Eosinophils play a variety of roles in the immune response. Red blood cells are about 7 to 8 micrometers in diameter, and a neutrophil is about 10 to 12 micrometers. Credit. Modification of work by Dr. David Saba. Introduction. The environment consists of numerous pathogens, which are agents, usually microorganisms, that cause diseases in their hosts. A host is the organism that is invaded and often harmed by a pathogen. Pathogens include bacteria, protists, fungi and other infectious organisms. We are constantly exposed to pathogens in food and water, on surfaces, and in the air. Mammalian immune systems evolved for protection from such pathogens. They are composed of an extremely diverse array of specialized cells and soluble molecules that coordinate a rapid and flexible defense system capable of providing protection from a majority of these disease agents. Components of the immune system constantly search the body for signs of pathogens. When pathogens are found, immune factors are mobilized to the site of an infection. The immune factors identify the nature of the pathogen, strengthen the corresponding cells and molecules to combat it efficiently, and then halt the immune response after the infection is cleared to avoid unnecessary host cell damage. The immune system can remember pathogens to which it has been exposed to create a more efficient response upon re-exposure. This memory can last several decades. Features of the immune system, such as pathogen identification, specific response, amplification, retreat, and remembrance are essential for survival against pathogens. The immune response can be classified as either innate or active. The innate immune response is always present and attempts to defend against all pathogens rather than focusing on specific ones. Conversely, the adaptive immune response stores information about past infections and mounts pathogen-specific defenses. Chapter Outline 42.1 Innate Immune Response 42.2 Adaptive Immune Response 42.3 Antibodies 42.4 Disruptions in the Immune System 42.1 Innate Immune Response. By the end of this section, you will be able to do the following: describe physical and chemical immune barriers, explain immediate and induced innate immune responses, discuss natural killer cells, describe major histocompatibility class I molecules, summarize how the proteins in a complement system function to destroy extracellular pathogens. The immune system comprises both innate and adaptive immune responses. Innate immunity occurs naturally because of genetic factors or physiology. It is not induced by infection or vaccination but works to reduce the workload for the adaptive immune response. Both the innate and adaptive levels of the immune response involve secreted proteins, receptor-mediated signaling, and intricate cell-to-cell communication. The innate immune system developed early in animal evolution, roughly 1 billion years ago, as an essential response to infection. Innate immunity has a limited number of specific targets. Any pathogenic threat triggers a consistent sequence of events that can identify the type of pathogen and either clear the infection independently or mobilize a highly specialized adaptive immune response. For example, tears and mucus secretions contain microbicidal factors. Physical and chemical barriers. Before any immune factors are triggered, the skin functions as a continuous, impassable barrier to potentially infectious pathogens. Pathogens are killed or inactivated on the skin by desiccation, drying out, and by the skin's acidity. In addition, beneficial microorganisms that coexist on the skin compete with invading pathogens, preventing infection. Regions of the body that are not protected by skin, such as the eyes and mucous membranes, have alternative methods of defense, such as tears and mucus secretions that trap and rinse away pathogens, and cilia in the nasal passages and respiratory tract that push the mucus with the pathogens out of the body. Throughout the body are other defenses, such as the low pH of the stomach, which inhibits the growth of pathogens, blood proteins that bind and disrupt bacterial cell membranes, and the process of urination, which flushes pathogens from the urinary tract. Despite these barriers, pathogens may enter the body through skin abrasions or punctures, or by collecting on mucosal surfaces in large numbers that overcome the mucus or cilia. Some pathogens have evolved specific mechanisms that allow them to overcome physical and chemical barriers. When pathogens do enter the body, the innate immune system responds with inflammation, pathogen engulfment, and secretion of immune factors and proteins. Pathogen recognition and infection may be intracellular or extracellular, depending on the pathogen. All viruses infect cells and replicate within those cells, intracellularly, whereas bacteria and other parasites may replicate intracellularly or extracellularly, depending on the species. The innate immune system must respond accordingly, by identifying the extracellular pathogen and/or by identifying host cells that have already been infected. When a pathogen enters the body, cells in the blood and lymph detect the specific pathogen-associated molecular patterns, PAMPs, on the pathogen surface. PAMPs are carbohydrate, polypeptide, and nucleic acid signatures that are expressed by viruses, bacteria, and parasites but which differ from molecules on host cells. The immune system has specific cells, described in Figure 42.2 and shown in Figure 42.3, with receptors that recognize these PAMPs. A macrophage is a large phagocytic cell that engulfs foreign particles and pathogens. Macrophages recognize PAMPs via complementary pattern recognition receptors, PRRs. PRRs are molecules on macrophages and dendritic cells which are in contact with the external environment. A monocyte is a type of white blood cell that circulates in the blood and lymph and differentiates into macrophages after it moves into infected tissue. Dendritic cells bind molecular signatures of pathogens and promote pathogen engulfment and destruction. Toll-like receptors, TLRs, are a type of PRR that recognizes molecules that are shared by pathogens but distinguishable from host molecules. TLRs are present in invertebrates as well as vertebrates, and appear to be one of the most ancient components of 1194 Chapter 42. The Immune System access for free at OpenStax.org. The immune system. TLRs have also been identified in the mammalian nervous system. Figure 42.2. The characteristics and location of cells involved in the innate immune system are described. Credit. Modification of work by NIH, 42.1. Innate immune response 1195. Figure 42.3. Cells of the blood include 1. Monocytes, 2. Lymphocytes, 3. Neutrophils, 4. Red blood cells, and 5. Platelets. Note the very similar morphologies of the leukocytes, 1, 2, 3. Credit. Modification of work by Bruce Wetzel, Harry Schaefer, NCI. Scale bar data from Matt Russell. Cytokine release effect. The binding of PRRs with PAMPs triggers the release of cytokines, which signal that a pathogen is present and needs to be destroyed along with any infected cells. A cytokine is a chemical messenger that regulates cell differentiation, form and function, proliferation, production, and gene expression to affect immune responses. At least 40 types of cytokines exist in humans that differ in terms of the cell type that produces them, the cell type that responds to them, and the changes they produce. One type of cytokine, interferon, is illustrated in Figure 42.4. One subclass of cytokines is the interleukin, IL, so named because they mediate interactions between leukocytes, white blood cells. Interleukins are involved in bridging the innate and adaptive immune responses. In addition to being released from cells after PAMP recognition, cytokines are released by the infected cells which bind to nearby uninfected cells and induce those cells to release cytokines, which results in a cytokine burst. A second class of early-acting cytokines is interferons, which are released by infected cells as a warning to nearby uninfected cells. One of the functions of an interferon is to inhibit viral replication. They also have other important functions, such as tumor surveillance. Interferons work by signaling neighboring uninfected cells to destroy RNA and reduce protein synthesis, signaling neighboring infected cells to undergo apoptosis, programmed cell death, and activating immune cells. In response to interferons, uninfected cells alter their gene expression, which increases the cell's resistance to infection. One effect of interferon-induced gene expression is a sharply reduced cellular protein synthesis. Virally infected cells produce more viruses by synthesizing large quantities of viral proteins. Thus, by reducing protein synthesis, a cell becomes resistant to viral infection. 1196 Chapter 42. The Immune System access for free at OpenStax.org. Figure 42.4. Interferons are cytokines that are released by a cell infected with a virus. Response of neighboring cells to interferon helps stem the infection. Phagocytosis and inflammation. The first cytokines to be produced are pro-inflammatory. That is, they encourage inflammation, the localized redness, swelling, heat, and pain that result from the movement of leukocytes and fluid through increasingly permeable capillaries to a site of infection. The population of leukocytes that arrives at an infection site depends on the nature of the infecting pathogen. Both macrophages and dendritic cells engulf pathogens and cellular debris through phagocytosis. A neutrophil is also a phagocytic leukocyte that engulfs and digests pathogens. Neutrophils, shown in Figure 42.3, are the most abundant leukocytes of the immune system. Neutrophils have a nucleus with 2 to 5 lobes, and they contain organelles, called lysosomes, that digest engulfed pathogens. An eosinophil is a leukocyte that works with other eosinophils to surround a parasite. It is involved in the allergic response and in protection against helminths, parasitic worms. Neutrophils and eosinophils are particularly important leukocytes that engulf large pathogens, such as bacteria and fungi. A mast cell is a leukocyte that produces inflammatory molecules, such as histamine, in response to large pathogens. A basophil is a leukocyte that, like a neutrophil, releases chemicals to stimulate the inflammatory response as illustrated in Figure 42.5. Basophils are also involved in allergy and hypersensitivity responses and induce specific types of inflammatory responses. Eosinophils and basophils produce additional inflammatory mediators to recruit more leukocytes. A hypersensitive immune response to harmless antigens, such as in pollen, often involves the release of histamine by basophils and mast cells. Figure 42.5. In response to a cut, mast cells secrete histamines that cause nearby capillaries to dilate. Neutrophils and monocytes leave the capillaries. Monocytes mature into macrophages. Neutrophils, dendritic cells, and macrophages release chemicals to stimulate the inflammatory response. Neutrophils and macrophages also consume invading bacteria by phagocytosis. Cytokines also send feedback to cells of the nervous system to bring about the overall symptoms of feeling sick, which include 42.1, innate immune response, 1197 lethargy, muscle pain, and nausea. These effects may have evolved because the symptoms encourage the individual to rest and prevent the spreading of the infection to others. Cytokines also increase the core body temperature, causing a fever, which causes the liver to withhold iron from the blood. Without iron, certain pathogens, such as some bacteria, are unable to replicate. This is called nutritional immunity. Link to learning. Watch this 23-second stop-motion video, http://openstax.org/l/canidia, showing a neutrophil that searches for and engulfs fungus spores during an elapsed time of about 79 minutes. Natural killer cells. Lymphocytes are leukocytes that are histologically identifiable by their large, darkly staining nuclei. They are small cells with very little cytoplasm, as shown in Figure 42.6. Infected cells are identified and destroyed by natural killer NK cells, lymphocytes that can kill cells infected with viruses or tumor cells, abnormal cells that uncontrollably divide and invade other tissue. T cells and B cells of the adaptive immune system also are classified as lymphocytes. T cells are lymphocytes that mature in the thymus gland, and B cells are lymphocytes that mature in the bone marrow. NK cells identify intracellular infections, especially from viruses, by the altered expression of major histocompatibility class (MHC) I molecules on the surface of infected cells. MHC I molecules are proteins on the surfaces of all nucleated cells, thus they are scarce on red blood cells and platelets, which are non-nucleated. The function of MHC I molecules is to display fragments of proteins from the infectious agents within the cell to T cells. Healthy cells will be ignored, while non-self or foreign proteins will be attacked by the immune system. MHC2 molecules are found mainly on cells containing antigens, non-self proteins, and on lymphocytes. MHC2 molecules interact with helper T cells to trigger the appropriate immune response, which may include the inflammatory response. Figure 42.6. Lymphocytes, such as NK cells, are characterized by their large nuclei that actively absorb right stain and therefore appear dark-colored under a microscope. An infected cell, or a tumor cell, is usually incapable of synthesizing and displaying MHC I molecules appropriately. The metabolic resources of cells infected by some viruses produce proteins that interfere with MHCI processing and/or trafficking to the cell surface. The reduced MHCI on host cells varies from virus to virus and results from active inhibitors being produced by the viruses. This process can deplete host MHCI molecules on the cell surface, which NK cells detect as unhealthy or abnormal, while searching for cellular MHCI molecules. Similarly, the dramatically altered gene expression of tumor cells leads to expression of extremely deformed or absent MHCI molecules that also signal unhealthy or abnormal. NK cells are always active. An interaction with normal, intact MHCI molecules on a healthy cell disables the killing sequence, and the NK cell moves on. After the NK cell detects an infected or tumor cell, its cytoplasm secretes granules comprised of perforin, a destructive protein that creates a pore in the target cell. Granzymes are released along with the perforin in the immunological synapse. A granzyme is a protease that digests cellular proteins and induces the target cell to undergo programmed cell death, or apoptosis. Phagocytic cells then digest the cell debris left behind. NK cells are constantly patrolling the body and are an effective mechanism for controlling potential infections and preventing cancer progression. 1198 Chapter 42. The Immune System Access for free at OpenStax.org. Complement. An array of approximately 20 types of soluble proteins, called a complement system, functions to destroy extracellular pathogens. Cells of the liver and macrophages synthesize complement proteins continuously. These proteins are abundant in the blood serum and are capable of responding immediately to infecting microorganisms. The complement system is so named because it is complementary to the antibody response of the adaptive immune system. Complement proteins bind to the surfaces of microorganisms and are particularly attracted to pathogens that are already bound by antibodies. Binding of complement proteins occurs in a specific and highly regulated sequence, with each successive protein being activated by cleavage and/or structural changes induced upon binding of the preceding proteins. After the first few complement proteins bind, a cascade of sequential binding events follows in which the pathogen rapidly becomes coated in complement proteins. Complement proteins perform several functions. The proteins serve as a marker to indicate the presence of a pathogen to phagocytic cells, such as macrophages and B cells, and enhance engulfment. This process is called opsonization. Certain complement proteins can combine to form attack complexes that open pores in microbial cell membranes. These structures destroy pathogens by causing their contents to leak, as illustrated in Figure 42.7. 42.1. Innate immune response. 1199 Figure 42.7. The classic pathway for the complement cascade involves the attachment of several initial complement proteins to an antibody-bound pathogen, followed by rapid activation and binding of many more complement proteins and the creation of destructive pores in the microbial cell envelope and cell wall. The alternate pathway does not involve antibody activation. Rather, C3 convertase spontaneously breaks down C3. Endogenous regulatory proteins prevent the complement complex from binding to host cells. Pathogens lacking these regulatory proteins are lysed. Credit. Modification of work by NIH. 42.2 Adaptive immune response. By the end of this section, you will be able to do the following. Explain adaptive immunity. Compare and contrast adaptive and innate immunity. Describe cell-mediated immune response and humoral immune response. Describe immune tolerance. The adaptive, or acquired, immune response takes days or even weeks to become established, much longer than the innate. 1200 Chapter 42. The Immune System Access for free at OpenStax.org. Response. However, adaptive immunity is more specific to pathogens and has memory. Adaptive immunity is an immunity that occurs after exposure to an antigen, either from a pathogen or a vaccination. This part of the immune system is activated when the innate immune response is insufficient to control an infection. In fact, without information from the innate immune system, the adaptive response could not be mobilized. There are two types of adaptive responses: the cell-mediated immune response, which is carried out by T cells, and the humoral immune response, which is controlled by activated B cells and antibodies. Activated T cells and B cells that are specific to molecular structures on the pathogen proliferate and attack the invading pathogen. Their attack can kill pathogens directly or secrete antibodies that enhance the phagocytosis of pathogens and disrupt the infection. Adaptive immunity also involves a memory to provide the host with long-term protection from reinfection with the same type of pathogen. On re-exposure, this memory will facilitate an efficient and quick response. Antigen-presenting cells, unlike NK cells of the innate immune system, B cells, B lymphocytes, are a type of white blood cell that gives rise to antibodies, whereas T cells, T lymphocytes, are a type of white blood cell that plays an important role in the immune response. T cells are a key component in the cell-mediated response, the specific immune response that utilizes T cells to neutralize cells that have been infected with viruses and certain bacteria. There are three types of T cells: cytotoxic, helper, and suppressor T cells. Cytotoxic T cells destroy virus-infected cells in the cell-mediated immune response, and helper T cells play a part in activating both the antibody and the cell-mediated immune responses. Suppressor T cells deactivate T cells and B cells when needed, and thus prevent the immune response from becoming too intense. An antigen is a foreign or non-self macromolecule that reacts with cells of the immune system. Not all antigens will provoke a response. For instance, individuals produce innumerable self-antigens and are constantly exposed to harmless foreign antigens, such as food proteins, pollen, or dust components. The suppression of immune responses to harmless macromolecules is highly regulated and typically prevents processes that could be damaging to the host, known as tolerance. The innate immune system contains cells that detect potentially harmful antigens, and then inform the adaptive immune response about the presence of these antigens. An antigen-presenting cell, APC, is an immune cell that detects, engulfs, and informs the adaptive immune response about an infection. When a pathogen is detected, these APCs will phagocytose the pathogen and digest it to form many different fragments of the antigen. Antigen fragments will then be transported to the surface of the APC, where they will serve as an indicator to other immune cells. Dendritic cells are immune cells that process antigen material. They are present in the skin, Langerhans cells, and the lining of the nose, lungs, stomach, and intestines. Sometimes a dendritic cell presents on the surface of other cells to induce an immune response, thus functioning as an antigen-presenting cell. Macrophages also function as APCs. Before activation and differentiation, B cells can also function as APCs. After phagocytosis by APCs, the phagocytic vesicle fuses with an intracellular lysosome forming phagolysosome. Within the phagolysosome, the components are broken down into fragments. The fragments are then loaded onto MHC class I or MHC class II molecules and are transported to the cell surface for antigen presentation, as illustrated in Figure 42.8. Note that T lymphocytes cannot properly respond to the antigen unless it is processed and embedded in an MHC2 molecule. APCs express MHC on their surfaces, and when combined with a foreign antigen, these complexes signal a non-self invader. Once the fragment of antigen is embedded in the MHC2 molecule, the immune cell can respond. Helper T cells are one of the main lymphocytes that respond to antigen-presenting cells. Recall that all other nucleated cells of the body expressed MHCI molecules, which signal healthy or normal. 42.2. Adaptive immune response 1201 Figure 42.8. An APC, such as a macrophage, engulfs and digests a foreign bacterium. An antigen from the bacterium is presented on the cell surface in conjunction with an MHC2 molecule. Lymphocytes of the adaptive immune response interact with antigen-embedded MHC2 molecules to mature into functional immune cells. Link to learning this animation, http://openstax.org/l/immune_system. From Rockefeller University shows how dendritic cells act as sentinels in the body's immune system. T and B lymphocytes. Lymphocytes in human circulating blood are approximately 80 to 90% T cells, shown in Figure 42.9, and 10 to 20% B cells. Recall that the T cells are involved in the cell-mediated immune response, whereas B cells are part of the humoral immune response. T cells encompass a heterogeneous population of cells with extremely diverse functions. Some T cells respond to APCs of the innate immune system, and indirectly induce immune responses by releasing cytokines. Other T cells stimulate B cells to prepare their own response. Another population of T cells detects APC signals and directly kills the infected cells. Other T cells are involved in suppressing inappropriate immune reactions to harmless or self-antigens. Figure 42.9. This scanning electron micrograph shows a T lymphocyte, which is responsible for the cell-mediated immune response. T cells are able to recognize antigens. Credit. Modification of work by NCI. Scale bar data from Matt Russell. T and B cells exhibit a common theme of recognition, binding of specific antigens via a complementary receptor, followed by activation and self-amplification, maturation to specifically bind to the particular antigen of the infecting pathogen. T and B lymphocytes are also similar in that each cell only expresses one type of antigen receptor. Any individual may possess a 1202 Chapter 42. The Immune System access for free at OpenStax.org. Population of T and B cells that together express a near-limitless variety of antigen receptors that are capable of recognizing virtually any infecting pathogen. T and B cells are activated when they recognize small components of antigens, called epitopes, presented by APCs, illustrated in Figure 42.10. Note that recognition occurs at a specific epitope rather than on the entire antigen. For this reason, epitopes are known as antigenic determinants. In the absence of information from APCs, T and B cells remain inactive, or naive, and are unable to prepare an immune response. The requirement for information from the APCs of innate immunity to trigger B cell or T cell activation illustrates the essential nature of the innate immune response to the functioning of the entire immune system. Figure 42.10. An antigen is a macromolecule that reacts with components of the immune system. A given antigen may contain several motifs that are recognized by immune cells. Each motif is an epitope. In this figure, the entire structure is an antigen, and the orange, salmon and green components projecting from it represent potential epitopes. Naive T cells can express one of two different molecules, CD4 or CD8, on their surface, as shown in Figure 42.11, and are accordingly classified as CD4+ or CD8+ cells. These molecules are important because they regulate how a T cell will interact with and respond to an APC. Naive CD4+ cells bind APCs via their antigen-embedded MHC2 molecules and are stimulated to become helper T, Th, lymphocytes, cells that go on to stimulate B cells, or cytotoxic T cells, directly or secrete cytokines to inform more and various target cells about the pathogenic threat. In contrast, CD8+ cells engage antigen-embedded MHCI molecules on APCs and are stimulated to become cytotoxic T lymphocytes, CTLs, which directly kill infected cells by apoptosis and emit cytokines to amplify the immune response. The two populations of T cells have different mechanisms of immune protection, but both bind MHC molecules via their antigen receptors called T cell receptors, TCRs. The CD4 or CD8 surface molecules differentiate whether the TCR will engage an MHC2 or an MHCI molecule. Because they assist in binding specificity, the CD4 and CD8 molecules are described as coreceptors. 42.2. Adaptive Immune Response 1203 Visual Connection Figure 42.11 Naive CD4 plus T cells engage MHC2 molecules on antigen-presenting cells, APCs, and become activated. Clones of the activated helper T cell, in turn, activate B cells and CD8 plus T cells, which become cytotoxic T cells. Cytotoxic T cells kill infected cells. Which of the following statements about T cells is false? A. Helper T cells release cytokines while cytotoxic T cells kill the infected cell. B. Helper T cells are CD4+, while cytotoxic T cells are CD8+. C. MHC2 is a receptor found on most body cells, while MHCI is a receptor found on immune cells only. D. The T cell receptor is found on both CD4+ and CD8+ T cells. Consider the innumerable possible antigens that an individual will be exposed to during a lifetime. The mammalian adaptive immune system is adept in responding appropriately to each antigen. Mammals have an enormous diversity of T cell populations, resulting from the diversity of TCRs. Each TCR consists of two polypeptide chains that span the T cell membrane, as illustrated in Figure 42.12. The chains are linked by a disulfide bridge. Each polypeptide chain is comprised of a constant domain and a variable domain. A domain, in this sense, is a specific region of a protein that may be regulatory or structural. The intracellular domain is involved in intracellular signaling. A single T cell will express thousands of identical copies of one specific TCR variant on its cell surface. The specificity of the adaptive immune system occurs because it synthesizes millions of different T cell populations, each expressing a TCR that differs in its variable domain. This TCR diversity is achieved by the mutation and recombination of genes that encode these receptors in stem cell precursors of T cells. The binding between an antigen displaying MHC molecule and a complementary TCR match indicates that the adaptive immune system needs to activate and produce that specific T cell because its structure is appropriate to recognize and destroy the invading pathogen. 1204 Chapter 42. The Immune System Access for free at OpenStax.org. Figure 42.12. A T cell receptor spans the membrane and projects variable binding regions into the extracellular space to bind processed antigens via MHC molecules on APCs. Helper T lymphocytes. The Th lymphocytes function indirectly to identify potential pathogens for other cells of the immune system. These cells are important for extracellular infections, such as those caused by certain bacteria, helminths, and protozoa. Th lymphocytes recognize specific antigens displayed in the MHC2 complexes of APCs. There are two major populations of Th cells, Th1 and Th2. Th1 cells secrete cytokines to enhance the activities of macrophages and other T cells. Th1 cells activate the action of cytotoxic T cells, as well as macrophages. Th2 cells stimulate naive B cells to destroy foreign invaders via antibody secretion. Whether a Th1 or a Th2 immune response develops depends on the specific types of cytokines secreted by cells of the innate immune system, which in turn depends on the nature of the invading pathogen. The Th1-mediated response involves macrophages and is associated with inflammation. Recall the frontline defenses of macrophages involved in the innate immune response. Some intracellular bacteria, such as Mycobacterium tuberculosis, have evolved to multiply in macrophages after they have been engulfed. These pathogens evade attempts by macrophages to destroy and digest the pathogen. When M. tuberculosis infection occurs, macrophages can stimulate naive T cells to become Th1 cells. These stimulated T cells secrete specific cytokines that send feedback to the macrophage to stimulate its digestive capabilities and allow it to destroy the colonizing M. tuberculosis. In the same manner, Th1-activated macrophages also become better suited to ingest and kill tumor cells. In summary, Th1 responses are directed toward intracellular invaders while Th2 responses are aimed at those that are extracellular. B lymphocytes. When stimulated by the Th2 pathway, naive B cells differentiate into antibody-secreting plasma cells. A plasma cell is an immune cell that secretes antibodies. These cells arise from B cells that were stimulated by antigens. Similar to T cells, naive B cells initially are coated in thousands of B cell receptors, BCRs, which are membrane-bound forms of Ig, immunoglobulin, or an antibody. The B cell receptor has two heavy chains and two light chains connected by disulfide linkages. Each chain has a constant and a variable region. The latter is involved in antigen binding. Two other membrane proteins, Ig-alpha and Ig-beta, are involved in signaling. The receptors of any particular B cell, as shown in Figure 42.13, are all the same, but the hundreds of millions of different B cells in an individual have distinct recognition domains that contribute to extensive diversity in the types of molecular structures to which they can bind. In this state, B cells function as APCs. They bind and engulf foreign antigens via their BCRs and then display processed antigens in the context of MHC2 molecules to Th2 cells. When a Th2 cell detects that a B cell is bound to a relevant antigen, it secretes specific cytokines that induce the B cell to proliferate rapidly, which makes thousands of identical, clonal, copies of it, and then it synthesizes and secretes antibodies with the same antigen recognition pattern as the BCRs. The activation of B cells corresponding to one specific BCR variant and the dramatic proliferation of that variant is known as clonal selection. This phenomenon drastically, but briefly, changes the proportions of BCR variants expressed by the immune system, and shifts the balance toward BCRs specific to the infecting pathogen. 42.2. Adaptive immune response 1205 Figure 42.13 B-cell receptors are embedded in the membranes of B cells and bind a variety of antigens through their variable regions. The signal transduction region transfers the signal into the cell. T and B cells differ in one fundamental way. Whereas T cells bind antigens that have been digested and embedded in MHC molecules by APCs, B cells function as APCs that bind intact antigens that have not been processed. Although T and B cells both react with molecules that are termed antigens, these lymphocytes actually respond to very different types of molecules. B cells must be able to bind intact antigens because they secrete antibodies that must recognize the pathogen directly, rather than digested remnants of the pathogen. Bacterial carbohydrate and lipid molecules can activate B cells independently from the T cells. Cytotoxic T lymphocytes (CTLs), a subclass of T cells, function to clear infections directly. The cell-mediated part of the adaptive immune system consists of CTLs that attack and destroy infected cells. CTLs are particularly important in protecting against viral infections. This is because viruses replicate within cells where they are shielded from extracellular contact with circulating antibodies. When APCs phagocytize pathogens and present MHCI-embedded antigens to naive CD8+ T cells that express complementary TCRs, the CD8+ T cells become activated to proliferate according to clonal selection. These resulting CTLs then identify non-APCs displaying the same MHCI-embedded antigens, for example, viral proteins. For example, the CTLs identify infected host cells. Intracellularly, infected cells typically die after the infecting pathogen replicates to a sufficient concentration and lyses the cell, as many viruses do. CTLs attempt to identify and destroy infected cells before the pathogen can replicate and escape, thereby halting the progression of intracellular infections. CTLs also support NK lymphocytes to destroy early cancers. Cytokines secreted by the Th1 response that stimulates macrophages also stimulate CTLs and enhance their ability to identify and destroy infected cells and tumors. CTLs sense MHCI-embedded antigens by directly interacting with infected cells via their TCRs. Binding of TCRs with antigens activates CTLs to release perforin and granzyme, degradative enzymes that will induce apoptosis of the infected cell. Recall that this is a similar destruction mechanism to that used by NK cells. In this process, the CTL does not become infected and is not harmed by the secretion of perforin and granzymes. In fact, the functions of NK cells and CTLs are complementary and maximize the removal of infected cells, as illustrated in Figure 42.14. If the NK cell cannot identify the missing self pattern of down-regulated MHCI molecules, then the CTL can identify it by the complex of MHCI with foreign antigens, which signals altered self. Similarly, if the CTL cannot detect antigen-embedded MHCI because the receptors are depleted from the cell surface, NK cells will destroy the cell instead. CTLs also emit cytokines, such as interferons, that alter surface protein expression in other infected cells, such that the infected cells can be easily identified and destroyed. Moreover, these interferons can also prevent virally infected cells from releasing virus particles. 1206 Chapter 42. The Immune System Access for free at OpenStax.org. Visual connection Figure 42.14 Natural killer. NK cells recognize the MHCI receptor on healthy cells. If MHCI is absent, the cell is lysed. Based on what you know about MHC receptors, why do you think an organ transplanted from an incompatible donor to a recipient will be rejected? Plasma cells and CTLs are collectively called effector cells. They represent differentiated versions of their naive counterparts, and they are involved in bringing about the immune defense of killing pathogens and infected host cells. Mucosal surfaces and immune tolerance. The innate and adaptive immune responses discussed thus far comprise the systemic immune system, affecting the whole body, which is distinct from the mucosal immune system. Mucosal immunity is formed by mucosa-associated lymphoid tissue, which functions independently of the systemic immune system, and which has its own innate and adaptive components. Mucosa-associated lymphoid tissue, MALT, illustrated in Figure 42.15, is a collection of lymphatic tissue that combines with epithelial tissue lining the mucosa throughout the body. This tissue functions as the immune barrier and response in areas of the body with direct contact to the external environment. The systemic and mucosal immune systems use many of the same cell types. Foreign particles that make their way to MALT are taken up by absorptive epithelial cells called M cells and delivered to APCs located directly below the mucosal tissue. M cells function in the transport described, and are located in the Peyer's patch, a lymphoid nodule. APCs of the mucosal immune system are primarily dendritic cells, with B cells and macrophages having minor roles. Processed antigens displayed on APCs are detected by T cells in the MALT and at various mucosal induction sites, such as the tonsils, adenoids, appendix, or the mesenteric lymph nodes of the intestine. Activated T cells then migrate through the lymphatic system and into the circulatory system to mucosal sites of infection. 42.2. Adaptive Immune Response 1207 Figure 42.15 The topology and function of intestinal MALT is shown. Pathogens are taken up by M cells in the intestinal epithelium and excreted into a pocket formed by the inner surface of the cell. The pocket contains antigen-presenting cells such as dendritic cells, which engulf the antigens, then present them with MHC2 molecules on the cell surface. The dendritic cells migrate to an underlying tissue called a Peyer's patch. Antigen-presenting cells, T cells, and B cells aggregate within the Peyer's patch, forming organized lymphoid follicles. There, some T cells and B cells are activated. Other antigen-loaded dendritic cells migrate through the lymphatic system where they activate B cells, T cells, and plasma cells in the lymph nodes. The activated cells then return to MALT tissue effector sites. IgA and other antibodies are secreted into the intestinal lumen. MALT is a crucial component of a functional immune system because mucosal surfaces, such as the nasal passages, are the first tissues onto which inhaled or ingested pathogens are deposited. The mucosal tissue includes the mouth, pharynx, and esophagus, and the gastrointestinal, respiratory, and urogenital tracts. The immune system has to be regulated to prevent wasteful, unnecessary responses to harmless substances, and more importantly so that it does not attack self. The acquired ability to prevent an unnecessary or harmful immune response to a detected foreign substance known not to cause disease is described as immune tolerance. Immune tolerance is crucial for maintaining mucosal homeostasis given the tremendous number of foreign substances, such as food proteins, that APCs of the oral cavity, pharynx, and gastrointestinal mucosa encounter. Immune tolerance is brought about by specialized APCs in the liver, lymph nodes, small intestine, and lung that present harmless antigens to an exceptionally diverse population of regulatory T, Treg, cells, specialized lymphocytes that suppress local inflammation and inhibit the secretion of stimulatory immune factors. The combined result of Treg cells is to prevent immunologic activation and inflammation in undesired tissue compartments and to allow the immune system to focus on pathogens instead. In addition to promoting immune tolerance of harmless antigens, other subsets of Treg cells are involved in the prevention of the autoimmune response, which is an inappropriate immune response to host cells or self-antigens. Another Treg class suppresses immune responses to harmful pathogens after the infection 1208 Chapter 42. The Immune System Access for Free at OpenStax.org. Has cleared to minimize host cell damage induced by inflammation and cell lysis. Immunological memory. The adaptive immune system possesses a memory component that allows for an efficient and dramatic response upon reinvasion of the same pathogen. Memory is handled by the adaptive immune system with little reliance on cues from the innate response. During the adaptive immune response to a pathogen that has not been encountered before, called a primary response, plasma cells secreting antibodies and differentiated T cells increase, then plateau over time. As B and T cells mature into effector cells, a subset of the naive populations differentiates into B and T memory cells with the same antigen specificities, as illustrated in Figure 42.16. A memory cell is an antigen-specific B or T lymphocyte that does not differentiate into effector cells during the primary immune response, but that can immediately become effector cells upon re-exposure to the same pathogen. During the primary immune response, memory cells do not respond to antigens and do not contribute to host defenses. As the infection is cleared and pathogenic stimuli subside, the effectors are no longer needed, and they undergo apoptosis. In contrast, the memory cells persist in the circulation. Visual connection figure 42.16. After initially binding an antigen to the B cell receptor, BCR, a B cell internalizes the antigen and presents it on MHC2. A helper T cell recognizes the MHC2 antigen complex and activates the B cell. As a result, memory B cells and plasma cells are made. The Rh antigen is found on Rh-positive red blood cells. An Rh-negative female can usually carry an Rh-positive fetus to term without difficulty. However, if she has a second Rh-positive fetus, her body may launch an immune attack that causes hemolytic 42.2, adaptive immune response 1209 disease of the newborn. Why do you think hemolytic disease is only a problem during the second or subsequent pregnancies? If the pathogen is never encountered again during the individual's lifetime, B and T memory cells will circulate for a few years or even several decades and will gradually die off, having never functioned as effector cells. However, if the host is re-exposed to the same pathogen type, circulating memory cells will immediately differentiate into plasma cells and CTLs without input from APCs or TH cells. One reason the adaptive immune response is delayed is because it takes time for naive B and T cells with the appropriate antigen specificities to be identified and activated. Upon reinfection, this step is skipped, and the result is a more rapid production of immune defenses. Memory B cells that differentiate into plasma cells output tens to hundreds-fold greater antibody amounts than were secreted during the primary response, as the graph in figure 42.17 illustrates. This rapid and dramatic antibody response may stop the infection before it can even become established, and the individual may not realize he or she had been exposed. Figure 42.17. In the primary response to infection, antibodies are secreted first from plasma cells. Upon re-exposure to the same pathogen, memory cells differentiate into antibody-secreting plasma cells that output a greater amount of antibody for a longer period of time. Vaccination is based on the knowledge that exposure to non-infectious antigens, derived from known pathogens, generates a mild primary immune response. The immune response to vaccination may not be perceived by the host as illness but still confers immune memory. When exposed to the corresponding pathogen to which an individual was vaccinated, the reaction is similar to a secondary exposure. Because each reinfection generates more memory cells and increased resistance to the pathogen, and because some memory cells die, certain vaccine courses involve one or more booster vaccinations to mimic repeat exposures. For instance, tetanus boosters are necessary every 10 years because the memory cells only live that long. Mucosal immune memory, a subset of T and B cells of the mucosal immune system, differentiates into memory cells just as in the systemic immune system. Upon reinvasion of the same pathogen type, a pronounced immune response occurs at the mucosal site where the original pathogen deposited, but a collective defense is also organized within interconnected or adjacent mucosal tissue. For instance, the immune memory of an infection in the oral cavity would also elicit a response in the pharynx if the oral cavity was exposed to the same pathogen. Career connection vaccinologist vaccination, or immunization, involves the delivery, usually by injection as shown in figure 42.18, of non-infectious antigens derived from known pathogens. Other components, called adjuvants, are delivered in parallel to help stimulate the immune response. Immunological memory is the reason vaccines work. Ideally, the effect of vaccination is to elicit immunological memory, and thus resistance to specific pathogens without the individual having to experience an infection. 1210 Chapter 42, The Immune System, access for free at OpenStax.org. Figure 42.18. Vaccines are often delivered by injection into the arm. Credit: U.S. Navy photographer's mate Airman Apprentice Christopher D. Blackley. Vaccinologists are involved in the process of vaccine development from the initial idea to the availability of the completed vaccine. This process can take decades, can cost millions of dollars, and can involve many obstacles along the way. For instance, injected vaccines stimulate the systemic immune system, eliciting humoral and cell-mediated immunity, but have little effect on the mucosal response, which presents a challenge because many pathogens are deposited and replicate in mucosal compartments, and the injection does not provide the most efficient immune memory for these disease agents. For this reason, vaccinologists are actively involved in developing new vaccines that are applied via intranasal, aerosol, oral, or transcutaneous, absorbed through the skin, delivery methods. Importantly, mucosal-administered vaccines elicit both mucosal and systemic immunity and produce the same level of disease resistance as injected vaccines. Figure 42.19. The polio vaccine can be administered orally. Credit. Modification of work by UNICEF's Virage. Currently, a version of intranasal influenza vaccine is available, and the polio and typhoid vaccines can be administered orally, as shown in Figure 42.19. Similarly, the measles and rubella vaccines are being adapted to aerosol delivery using inhalation devices. Eventually, transgenic plants may be engineered to produce vaccine antigens that can be eaten to confer disease resistance. Other vaccines may be adapted to rectal or vaginal application to elicit immune responses in rectal, genitourinary, or reproductive mucosa. Finally, vaccine antigens may be adapted to transdermal application in which the skin is lightly scraped and microneedles are used to pierce the outermost layer. In addition to mobilizing the mucosal immune response, this new generation of vaccines may end the anxiety associated with injections and, in turn, improve patient participation. 42.2. Adaptive immune response. 1211 Primary centers of the immune system. Although the immune system is characterized by circulating cells throughout the body, the regulation, maturation, and intercommunication of immune factors occur at specific sites. The blood circulates immune cells, proteins, and other factors through the body. Approximately 0.1% of all cells in the blood are leukocytes, which encompass monocytes, the precursor of macrophages, and lymphocytes. The majority of cells in the blood are erythrocytes, red blood cells. Lymph is a watery fluid that bathes tissues and organs with protective white blood cells and does not contain erythrocytes. Cells of the immune system can travel between the distinct lymphatic and blood circulatory systems, which are separated by interstitial space, by a process called extravasation, passing through to surrounding tissue. The cells of the immune system originate from hematopoietic stem cells in the bone marrow. Cytokines stimulate these stem cells to differentiate into immune cells. B-cell maturation occurs in the bone marrow, whereas naive T cells transit from the bone marrow to the thymus for maturation. In the thymus, immature T cells that express TCRs complementary to self-antigens are destroyed. This process helps prevent autoimmune responses. On maturation, T and B lymphocytes circulate to various destinations. Lymph nodes scattered throughout the body, as illustrated in Figure 42.20, house large populations of T and B cells, dendritic cells, and macrophages. Lymph gathers antigens as it drains from tissues. These antigens then are filtered through lymph nodes before the lymph is returned to circulation. APCs in the lymph nodes capture and process antigens and inform nearby lymphocytes about potential pathogens. Figure 42.20. Lymphatic vessels carry a clear fluid called lymph throughout the body. The liquid enters lymph nodes through afferent vessels. Lymph nodes are filled with lymphocytes that purge infecting cells. The lymph then exits through efferent vessels. Credit. Modification of work by NIH, NCI. The spleen houses B and T cells, macrophages, dendritic cells, and NK cells. The spleen, shown in Figure 42.21, is the site where APCs that have trapped foreign particles in the blood can communicate with lymphocytes. Antibodies are synthesized and secreted by activated plasma cells in the spleen, and the spleen filters foreign substances and antibody-complexed pathogens from the blood. Functionally, the spleen is to the blood as lymph nodes are to the lymph. 1212 Chapter 42. The Immune System Access for free at OpenStax.org. Figure 42.21. The spleen is similar to a lymph node but is much larger and filters blood instead of lymph. Blood enters the spleen through arteries and exits through veins. The spleen contains two types of tissue, red pulp and white pulp. Red pulp consists of cavities that store blood. Within the red pulp, damaged red blood cells are removed and replaced by new ones. White pulp is rich in lymphocytes that remove antigen-coated bacteria from the blood. Credit. Modification of work by NCI. 42.3 Antibodies. By the end of this section, you will be able to do the following. Explain cross-reactivity. Describe the structure and function of antibodies. Discuss antibody production. An antibody, also known as an immunoglobulin, Ig, is a protein that is produced by plasma cells after stimulation by an antigen. Antibodies are the functional basis of humoral immunity. Antibodies occur in the blood, in gastric and mucus secretions, and in breast milk. Antibodies in these bodily fluids can bind pathogens and mark them for destruction by phagocytes before they can infect cells. Antibody structure. An antibody molecule is comprised of 4 polypeptides, 2 identical heavy chains, large peptide units, that are partially bound to each other in a Y formation, which are flanked by 2 identical light chains, small peptide units, as illustrated in Figure 42.22. Bonds between the cysteine amino acids in the antibody molecule attach the polypeptides to each other. The areas where the antigen is recognized on the antibody are variable domains, and the antibody base is composed of constant domains. In germline B cells, the variable region of the light chain gene has 40 variable, V, and 5 joining, J, segments. An enzyme called DNA recombinase randomly excises most of these segments out of the gene, and splices one V segment to one J segment. During RNA processing, all but one V and J segment are spliced out. Recombination and splicing may result in over 106 possible VJ combinations. As a result, each differentiated B cell in the human body typically has a unique variable chain. The constant domain, which does not bind antibody, is the same for all antibodies. 42.3. Antibodies. 1213 Figure 42.22, As a germline B cell matures, an enzyme called DNA recombinase randomly excises V and J segments from the light chain gene. Splicing at the mRNA level results in further gene rearrangement. As a result, each antibody has a unique variable region capable of binding a different antigen. Similar to TCRs and BCRs, antibody diversity is produced by the mutation and recombination of approximately 300 different gene segments encoding the light and heavy chain variable domains in precursor cells that are destined to become B cells. The variable domains from the heavy and light chains interact to form the binding site through which an antibody can bind a specific epitope on an antigen. The numbers of repeated constant domains in Ig classes are the same for all antibodies corresponding to a specific class. Antibodies are structurally similar to the extracellular component of the BCRs, and B-cell maturation to plasma cells can be visualized in simple terms as the cell acquires the ability to secrete the extracellular portion of its BCR in large quantities. Antibody classes. Antibodies can be divided into 5 classes: IgM, Ig, IgA, IgD, IgE, based on their physiochemical, structural, and immunological properties. IgGs, which make up about 80% of all antibodies, have heavy chains that consist of 1 variable domain and 3 identical constant domains. IgA and IgD also have 3 constant domains per heavy chain, whereas IgM and IgE each have 4 constant domains per heavy chain. The variable domain determines binding specificity, and the constant domain of the heavy chain determines the immunological mechanism of action of the corresponding antibody class. It is possible for 2 antibodies to have the same binding specificities but be in different classes and, therefore, to be involved in 1214 Chapter 42, the immune system. Access for free at OpenStax.org. Different functions. After an adaptive defense is produced against a pathogen, typically plasma cells first secrete IgM into the blood. BCRs on naive B cells are of the IgM class and occasionally IgD class. IgM molecules make up approximately 10% of all antibodies. Prior to antibody secretion, plasma cells assemble IgM molecules into pentamers, 5 individual antibodies, linked by a joining J chain, as shown in Figure 42.23. The pentamer arrangement means that these macromolecules can bind 10 identical antigens. However, IgM molecules released early in the adaptive immune response do not bind to antigens as stably as IgGs, which are one of the possible types of antibodies secreted in large quantities upon re-exposure to the same pathogen. Figure 42.23 summarizes the properties of immunoglobulins and illustrates their basic structures. Figure 42.23 immunoglobulins have different functions, but all are composed of light and heavy chains that form a Y-shaped structure. IgAs populate the saliva, tears, breast milk, and mucus secretions of the gastrointestinal, respiratory, and genitourinary tracts. Collectively, these bodily fluids coat and protect the extensive mucosa, 4,000 square feet in humans. The total number of IgA molecules in these bodily secretions is greater than the number of Ig molecules in the blood serum. A small amount of IgA is also secreted into the serum in monomeric form. Conversely, some IgM is secreted into bodily fluids of the mucosa. Similar to IgM, IgA molecules are secreted as polymeric structures linked with a J-chain. However, IgAs are secreted mostly as dimeric molecules, not pentamers. IgE is present in the serum in small quantities and is best characterized in its role as an allergy mediator. IgD is also present in small quantities. Similar to IgM, BCRs of the IgD class are found on the surface of naive B cells. This class supports antigen recognition and maturation of B cells to plasma cells. Antibody functions differentiated plasma cells are crucial players in the humoral response, and the antibodies they secrete are particularly significant against extracellular pathogens and toxins. Antibodies circulate freely and act independently of plasma cells. Antibodies can be transferred from one individual to another to temporarily protect against infectious disease. For instance, a person who has recently produced a successful immune response against a particular disease agent can donate blood to a non-immune recipient and confer temporary immunity through antibodies in the donor's blood serum. This phenomenon is called passive immunity. It also occurs naturally during breastfeeding, which makes breastfed infants highly resistant to infections during the first few months of life. Antibodies coat extracellular pathogens and neutralize them, as illustrated in Figure 42.24. By blocking key sites on the pathogen that enhance their infectivity, such as receptors that dock pathogens on host cells. Antibody neutralization can prevent pathogens from entering and infecting host cells, as opposed to the CTL-mediated approach of killing cells that are already infected to prevent progression of an established infection. The neutralized antibody-coated pathogens can then be filtered by the spleen and eliminated in urine or feces. 42.3. Antibodies 1215 Figure 42.24 Antibodies may inhibit infection by a. Preventing the antigen from binding its target, b. Tagging a pathogen for destruction by macrophages or neutrophils, or c. Activating the complement cascade. Antibodies also mark pathogens for destruction by phagocytic cells, such as macrophages or neutrophils, because phagocytic cells are highly attracted to macromolecules complexed with antibodies. Phagocytic enhancement by antibodies is called opsonization. In a process called complement fixation, IgM and Ig in serum bind to antigens and provide docking sites onto which sequential complement proteins can bind. The combination of antibodies and complement enhances opsonization even further and promotes rapid clearing of pathogens. Affinity, avidity, and cross-reactivity. Not all antibodies bind with the same strength, specificity, and stability. In fact, antibodies exhibit different affinities, attraction, depending on the molecular complementarity between antigen and antibody molecules, as illustrated in Figure 42.25. An antibody with a higher affinity for a particular antigen would bind more strongly and stably, and thus would be expected to present a more challenging defense against the pathogen corresponding to the specific antigen. 1216 Chapter 42. The Immune System Access for free at OpenStax.org. Figure 42.25. Affinity refers to the strength of single interaction between antigen and antibody, while avidity refers to the strength of all interactions combined. b. An antibody may cross-react with different epitopes. The term avidity describes binding by antibody classes that are secreted as joined, multivalent structures, such as IgM and IgA. Although avidity measures the strength of binding, just as affinity does, the avidity is not simply the sum of the affinities of the antibodies in a multimeric structure. The avidity depends on the number of identical binding sites on the antigen being detected, as well as other physical and chemical factors. Typically, multimeric antibodies, such as pentameric IgM, are classified as having lower affinity than monomeric antibodies, but high avidity. Essentially, the fact that multimeric antibodies can bind many antigens simultaneously balances their slightly lower binding strength for each antibody-antigen interaction. Antibodies secreted after binding to one epitope on an antigen may exhibit cross-reactivity for the same or similar epitopes on different antigens. Because an epitope corresponds to such a small region, the surface area of about 4 to 6 amino acids, it is possible for different macromolecules to exhibit the same molecular identities and orientations over short regions. Cross-reactivity describes when an antibody binds not to the antigen that elicited its synthesis and secretion, but to a different antigen. Cross-reactivity can be beneficial if an individual develops immunity to several related pathogens despite having only been exposed to or vaccinated against one of them. For instance, antibody cross-reactivity may occur against the similar surface structures of various gram-negative bacteria. Conversely, antibodies raised against pathogenic molecular components that resemble self-molecules may incorrectly mark host cells for destruction and cause autoimmune damage. Patients who develop systemic lupus erythematosus, SLE, commonly exhibit antibodies that react with their own DNA. These antibodies may have been initially raised against the nucleic acid of microorganisms but later cross-reacted with self-antigens. This phenomenon is also called molecular mimicry. 42.3. Antibodies 1217 Antibodies of the mucosal immune system. Antibodies synthesized by the mucosal immune system include IgA and IgM. Activated B cells differentiate into mucosal plasma cells that synthesize and secrete dimeric IgA, and to a lesser extent, pentameric IgM. Secreted IgA is abundant in tears, saliva, breast milk, and in secretions of the gastrointestinal and respiratory tracts. Antibody secretion results in a local humoral response at epithelial surfaces and prevents infection of the mucosa by binding and neutralizing pathogens. 42.4 Disruptions in the immune system. By the end of this section, you will be able to do the following. Describe hypersensitivity, define autoimmunity. A functioning immune system is essential for survival, but even the sophisticated cellular and molecular defenses of the mammalian immune response can be defeated by pathogens at virtually every step. In the competition between immune protection and pathogen evasion, pathogens have the advantage of more rapid evolution because of their shorter generation time and other characteristics. For instance, Streptococcus pneumoniae, bacterium that cause pneumonia and meningitis, surrounds itself with a capsule that inhibits phagocytes from engulfing it and displaying antigens to the adaptive immune system. Staphylococcus aureus, bacterium that can cause skin infections, abscesses, and meningitis, synthesizes a toxin called leukocidin that kills phagocytes after they engulf the bacterium. Other pathogens can also hinder the adaptive immune system. HIV infects Th cells via their CD4 surface molecules, gradually depleting the number of Th cells in the body. This inhibits the adaptive immune system's capacity to generate sufficient responses to infection or tumors. As a result, HIV-infected individuals often suffer from infections that would not cause illness in people with healthy immune systems but which can cause devastating illness to immune-compromised individuals. Maladaptive responses of immune cells and molecules themselves can also disrupt the proper functioning of the entire system, leading to host cell damage that could become fatal. Immunodeficiency failures, insufficiencies, or delays at any level of the immune response can allow pathogens or tumor cells to gain a foothold and replicate or proliferate to high enough levels that the immune system becomes overwhelmed. Immunodeficiency is the failure, insufficiency, or delay in the response of the immune system, which may be acquired or inherited. Immunodeficiency can be acquired as a result of infection with certain pathogens, such as HIV, chemical exposure, including certain medical treatments, malnutrition, or possibly by extreme stress. For instance, radiation exposure can destroy populations of lymphocytes and elevate an individual's susceptibility to infections and cancer. Dozens of genetic disorders result in immunodeficiencies, including severe combined immunodeficiency, SCID, Bayer lymphocyte syndrome, and MHC2 deficiencies. Rarely, primary immunodeficiencies that are present from birth may occur. Neutropenia is one form in which the immune system produces a below-average number of neutrophils, the body's most abundant phagocytes. As a result, bacterial infections may go unrestricted in the blood, causing serious complications. Hypersensitivity's maladaptive immune responses toward harmless foreign substances or self-antigens that occur after tissue sensitization are termed hypersensitivities. The types of hypersensitivities include immediate, delayed, and autoimmunity. A large proportion of the population is affected by one or more types of hypersensitivity. Allergies: the immune reaction that results from immediate hypersensitivities in which an antibody-mediated immune response occurs within minutes of exposure to a harmless antigen is called an allergy. In the United States, 20% of the population exhibits symptoms of allergy or asthma, whereas 55% test positive against one or more allergens. Upon initial exposure to a potential allergen, an allergic individual synthesizes antibodies of the IgE class via the typical process of APCs presenting processed antigen to Th cells that stimulate B cells to produce IgE. This class of antibodies also mediates the immune response to parasitic worms. The constant domain of the IgE molecules interact with mast cells embedded in connective tissues. This process primes, or sensitizes, the tissue. Upon subsequent exposure to the same allergen, IgE molecules on mast cells bind the antigen via their variable domains and stimulate the mast cell to release the modified amino acids histamine and serotonin. These chemical mediators then recruit eosinophils which mediate allergic responses. Figure 42.26 shows an example of an allergic response to ragweed pollen. The effects of an allergic reaction range from mild symptoms like sneezing and itchy, watery eyes to more severe or even life-threatening reactions involving intensely itchy welts or hives, airway contraction with severe 12-18 Chapter 42. The Immune System access for free at OpenStax.org. Respiratory distress and plummeting blood pressure. This extreme reaction is known as anaphylactic shock. If not treated with epinephrine to counter the blood pressure and breathing effects, this condition can be fatal. Figure 42.26 On first exposure to an allergen, an IgE antibody is synthesized by plasma cells in response to a harmless antigen. The IgE molecules bind to mast cells, and on secondary exposure, the mast cells release histamines and other modulators that affect the symptoms of allergy. Credit. Modification of work by NIH. Delayed hypersensitivity is a cell-mediated immune response that takes approximately 1 to 2 days after secondary exposure for a maximal reaction to be observed. This type of hypersensitivity involves the Th1 cytokine-mediated inflammatory response and may manifest as local tissue lesions or contact dermatitis, rash or skin irritation. Delayed hypersensitivity occurs in some individuals in response to contact with certain types of jewelry or cosmetics. Delayed hypersensitivity facilitates the immune response to poison ivy and is also the reason why the skin test for tuberculosis results in a small region of inflammation on individuals who were previously exposed to Mycobacterium tuberculosis. That is also why cortisone is used to treat such responses. It will inhibit cytokine production. Autoimmunity is a type of hypersensitivity to self-antigens that affects approximately 5% of the population. Most types of autoimmunity involve the humoral immune response. Antibodies that inappropriately mark self-components as foreign are termed autoantibodies. In patients with the autoimmune disease myasthenia gravis, muscle cell receptors that induce contraction in response to acetylcholine are targeted by antibodies. The result is muscle weakness that may include marked difficulty with fine and/or gross motor functions. In systemic lupus erythematosus, a diffuse autoantibody response to the individual's own DNA and proteins results in various systemic diseases. As illustrated in Figure 42.27, systemic lupus erythematosus may affect the heart, joints, lungs, skin, kidneys, central nervous system, or other tissues, causing tissue damage via antibody binding, complement recruitment, lysis, and inflammation. 42.4. Disruptions in the immune system. 1219 Figure 42.27 Systemic lupus erythematosus is characterized by autoimmunity to the individual's own DNA and/or proteins, which leads to varied dysfunction of the organs. Credit. Modification of work by Michael Hagstrom. Autoimmunity can develop with time, and its causes may be rooted in molecular mimicry. Antibodies and TCRs may bind self-antigens that are structurally similar to pathogen antigens, which the immune receptors first raised. As an example, infection with Streptococcus pyogenes, bacterium that causes strep throat, may generate antibodies or T cells that react with heart muscle, which has a similar structure to the surface of S. pyogenes. These antibodies can damage heart muscle with autoimmune attacks, leading to rheumatic fever. Insulin-dependent, type 1, diabetes mellitus arises from a destructive inflammatory Th1 response against insulin-producing cells of the pancreas. Patients with this autoimmunity must be injected with insulin that originates from other sources.